So we just finished up talking about the Hardy-Weinberg assumption that mating occurs at random. So this means if random mating occurs, then we are in Hardy-Weinberg equilibrium. If non-random mating occurs, we are changing genotype frequencies only. We're not removing any alleles from the population. We're not changing the allele frequency. We are simply changing the genotype frequency. So essentially we are affecting the number of heterozygotes in a population, but all of the alleles remain. So we went through random mating. Now we're going to go through the other 4 parts of the Hardy-Weinberg assumptions. We'll talk about mutation, migration, population size, and selection. Again, make sure that you know all the assumptions, make sure you know what the violation is, and make sure you know how it affects the population's allele frequency. All right, so let's dive in. So mutations affect allele frequencies. They do this by causing an increase in one and a decrease in another. So here our diagram is showing the change from the blue allele to a yellow allele. So we can think of this as blue being a functional mutation to a non-functional mutation. So individuals are normal or they have a phenotype that is somehow dysfunctional. So there are different ways you can have mutations. You can have mutations where you switch from one allele to another, blue to yellow. So allele 1 to allele 2. Or we can go the other way. We can go from yellow to blue, or allele 2 to allele 1. Now, if we go from blue to yellow, blue is going to decrease while yellow increases, and vice versa. So keep in mind that if one goes up, the other must go down, because P plus Q equals 1. If you increase one, you have to decrease the other. We can have a second situation where we produce a new allele. Sorry about that. So we could have the blue allele turn into a new red allele, in which case we would have blue, yellow, and red. So the introduction of that red is going to increase that allele in the population and decrease the other two for the same reason. P plus Q plus R equals 1. If we bring in another allele frequency, if any one of them goes up, the other two have to go down. Mutation, though, is generally the least likely to affect a population, because mutation rate is so low, especially if it's a large population. I believe I said this in the last video. If you have a large population and one person has that allele and then they don't pass it on, then there's no effect at all. But the chances of having a mutation and having a mutation in the gene that you are studying is incredibly low. So it's very unlikely that mutation rate is actually going to change the allele frequency, unless you have other things going on. So mutation rate in combination with some of these other Hardy-Weinberg assumptions can affect the population. So if you have a mutation and then you have limited migration, so no one's coming in or no one's going out, that mutation can eventually build up in a population. Or if you have a small population size and the individuals are forced into inbreeding, even if it's not close inbreeding, you're going to keep that mutation in the population. Or you may have a mutation that is very beneficial or very disadvantageous. And so then you have individuals who have these alleles that either produce more offspring or a lot less offspring, and you can cause a change due to mutation. But as I said, it's generally because these other things are going on. And then, of course, non-random mating is going to increase your chances. If you have a population that's inbreeding, it's going to increase your chances of having that allele. So here's an example of mutation combined with non-random mating. This is a condition called methemoglobinemia. So this is a fancy word to say that your blood has a blue tinge. So this is a disorder that is common in families in rural Kentucky. So historically, this was a region that was isolated from other populations. And because of that isolation, they tended to marry within families or within nearby families. So there was a lot of isolation. There was not a lot of intermarriage within people outside, between people outside of their population. So methemoglobinemia appeared as a mutation in one male, and after 2 to 3 generations, it began to appear more frequently. So you can see all of these cases of inbreeding, closely related inbreeding, between individuals who are relatives, and it essentially concentrated the allele within that family. So this disorder produces a form of hemoglobin where the iron molecule in the middle doesn't bind oxygen effectively. So this is actually something that happens normally. And normally about 1% of the hemoglobin in your blood appears blue because the iron essentially hasn't, or the oxygen hasn't been converted back into a form that is going to bind to the iron in your hemoglobin. And basically this happens as part of your process of dropping your oxygen off and picking it back up and moving it about. But individuals with this disorder have the inability to convert the methemoglobin to hemoglobin, and so they appear to be blue. So this can also be caused by some other environmental agents, some other drugs, but the outward effect is that the skin has a blue tinge. So this came to doctors' attention, I believe it was in the 60s, when individuals who did not know that they were related had a child together who appeared to be blue. And then doing some research into the family history, they found that they were in fact related, and this child had this allele that was identical by descent from this first individual that had this mutation. So you can see through this family tree that the mutation came up and then increased in frequency because of those consanguineous marriages. So this is kind of an interesting story. If you're interested, you can Google the blue fugates of Kentucky, and you can see pictures of the individuals. This is actually a treatable condition, and actually the individuals don't really have many effects of this disorder. So let's move on to our next Hardy-Weinberg assumption. The assumption is that there is no migration. When that is violated, migration causes allele frequencies to change. So this makes sense, right? If you have a population that have particular allele frequencies and individuals from another population come in at random with respect to the allele that you're looking at, meaning that they're not coming in because of that allele, they're just bringing the allele in by chance. You can probably understand how this is going to change allele frequencies. The impact of that movement is going to depend on the amount of difference between the populations. If the populations have very similar allele frequencies, they're not going to change very much by the migration of individuals. If they have drastically different allele frequencies, then the gaining population is going to change in the direction of the frequencies that are coming in. So if you have a group that has a very high frequency, they're going to increase the frequency of that particular allele. Migration also affects based on the number of migrants compared to the population size. So if you have a huge population size but only a small number of individuals coming in, they're probably going to have a negligible effect on your population. Now, this migration can also be out. So if individuals leave the population, they're taking their alleles with them. So they might leave at the same frequencies as they are in the population, or they might not. This is one of those where you can't really predict the direction of the change because it's sort of random which individuals are going to leave with respect to the allele that you are looking at. So this diagram here shows if we prevent migration between two populations, those populations can change. So if one population splits into two populations and they remain isolated from one another, there's no migration in between them, they're going to take their own evolutionary trajectory, and the allele frequencies are going to change based on selection for either of those populations based on genetic drift. So when you prevent migration, you're actually allowing the gene pools to become different. And those populations are going to diverge. If migration is allowed between two populations, you actually tend to make the populations more similar. So if two populations, these two here, are exchanging their information on their genetic information on a regular basis, then their allele frequencies are going to become more and more similar. So if we start out with one allele frequency and another allele frequency, and individuals combine into one population, some individuals come in, we actually are going to see sort of an average of the two. Again, this depends on the amount of migration. If it's just a little bit, it's going to have less of an effect. If there are more individuals, it's going to have greater effect. If these two populations are sharing their genetic information, they are not going to diverge. So they are going to remain more genetically similar. So if we prevent migration, if this happens for too long, those gene pools can eventually become so different that we get new species. And each one could become a subspecies, or each one could become fully their own species. Whereas if we allow migration, because they're remaining similar, we're probably not going to get a speciation event. Next Hardy-Weinberg assumption is that the population is infinitely large. So the violation here is that the population is not large and genetic drift or chance changes in allele frequencies can occur. So large is sort of relative. Infinitely large just means that chance events aren't going to affect the population. So depending on the population itself, we would have a different definition of infinitely large. But basically, we need to be large enough that every population is a sample, a good representation of the generation before. Genetic drift is where just by chance some individuals do not reproduce and leave their genes for the next generation. Now, this could be something like a natural disaster that wipes out individuals because they were in the wrong place at the wrong time. It might be that you're a plant and somebody plows through the field and kills all of the plants with the purple flowers. Really anything that is a chance event, something that is not related to the specific allele that you have. This can also just be just chance. Maybe some poor squirrel just doesn't find a mate that year, or maybe another squirrel finds lots of mates. So it really is anything that changes the allele frequencies in an unpredictable way. We say population is not infinitely large because chance events tend to affect small populations more greatly. So small populations are important here. So if we think about this in terms of flipping a coin, let's say we flip a coin 10 times. Just by chance, you might flip 3 heads and 7 tails, just by chance. It's unlikely because most likely you're going to get 50-50. Now, let's say we flip a coin 1,000 times. It's really unlikely that you're going to get 300 heads and 700 tails. You're more likely to get 50-50 because you have so many chances, and each individual chance is 50-50. It's the same thing for a population. When you have a small population, it is more likely that chance events are going to skew or force your allele frequencies into a particular direction. So there are 2 types of extreme genetic drift that we're going to talk about. One is the bottleneck effect. So the bottleneck effect is where you have a large population that in a short amount of time suddenly becomes a small population. So there are lots of animals that fall into this category: cheetahs, mountain lions, elephant seals. A lot of organisms fall into this bottleneck. So in a short amount of time, you lose a large number of population. And just by chance, because those individuals are lost regardless of the alleles that they have, just by chance, the allele frequencies are different from one generation to the next. So our diagram here depicts going from p-value of 0.7 to a p-value of 0.8, just by chance, because so many individuals are lost in a short amount of time. Then, if that population recovers and grows into its own later generation larger population, and Hardy-Weinberg assumptions are not being violated, we're going to maintain that 0.8. So just by chance, our population has fluctuated from 0.7 to 0.8. Now, this is also important because bottlenecks can also cause fixation, which is a loss. It is only one allele present. One allele becomes the only allele present. It becomes fixed in the population. Everyone in the population has that allele. And that is due to the chance loss of another allele. But then let's say that this small population doesn't become a large population. Let's say that it stays small. Now we can end up with our extinction vortex because this small population is going to be subject to inbreeding. And we're starting to lose genetic diversity. There's a greater chance that genetic drift is going to continue to impact this, and by chance, we might lose that Q allele, in which case our population will be fixed with the P allele. So the smaller your population gets, the more likely you're going to affect allele frequencies and the presence of alleles in general. I think that I just went through all of this on the last slide. So the bottleneck effect is just a chance event where a large portion of the population is wiped out regardless of their genotype, regardless of their alleles. So because so many individuals die in a short period of time, the next generation might have different allele frequencies. And then, of course, your remaining population is probably going to lack diversity. And this is a problem because if they are lacking genetic diversity, they are less likely going to be able to respond to new environmental pressures. And this is why we end up with endangered species, those that are in danger of extinction, because they have had some event, whether it is human-induced or environmental or whatever, they have had some event that has caused their population to decline. And that leads to a lack of genetic diversity, a loss of genetic diversity. So cheetahs are a fantastic example of this. Their range in Africa has been reduced by agriculture and building roads and just general encroachment of the human population. Cheetahs have been through so many bottlenecks that they basically are all identical. They show almost no polymorphism, almost no heterozygosity in their populations. They are so closely related that cheetahs in captivity can accept skin and tissue transplants from any other cheetah in captivity. Elephant seals are another example of this. In the late 1800s, they were hunted to near extinction. By 1900, there were only about 30 left. The current population has risen a bit, but individuals that have been studied are homozygous for all alleles that have been studied. So essentially, both of these populations have zero genetic diversity. This makes them more vulnerable to pathogens, and it also increases their rate of sterility. So genetic drift's effects are unpredictable. You cannot predict a chance event. You cannot predict a random fluctuation, but those chance events can have a large impact on a population. So this graph at the top shows a computer simulation of genetic drift. So just random changes in the allele frequencies. So they started out with two alleles, both at a frequency of 0.5 and a population size of 100. And then they just sort of let the simulation go. And you can see that there are 5 different scenarios here, 2 of which actually end up in fixation of one of the alleles over 100 generations. The other 2 show changes, just random fluctuations, and neither one is lost, but all 3 end up with different allele frequencies. And this is without any other impact of the other Hardy-Weinberg assumptions. The graph on the bottom shows the sort of the way that the alleles behaved if they looked at population number. In about 9% of populations, the allele went extinct, which means it was lost. And in about 8% of populations, one of the alleles became fixed over the other. I'm not sure why there's a discrepancy, because if you lose one, you fix the other. But this shows from 50%, we started out at 50%. These are the way that 300 different simulations where their final allele frequencies ended up. So this is all computer simulations, but it is a good visual on how drift can affect a population in very large ways, and how those changes are at random. It just squiggles on a line is how each generation changes. Another type of genetic drift is the founder effect. So the founder effect is when a small population basically establishes a new population. So a small population establishes a small group of individuals establishes a new population and expands into a larger area. Now, this is basically establishing a new group, and as part of this founder effect, they are isolated from the original population. So you might think about, a group of people moving to an island. So they're isolated from their founding population geographically, culturally, linguistically, from their surrounding population. So it doesn't have to be an island. It can be an island within a country. It can be, excuse me, a group of people who are somehow isolated from the population around them. Now, the founder effect says that by chance, allele frequencies differ between the founding population and the parent population. So the new population is going to have a different allele frequency than the founding population just by chance. So the founder effect has had a major impact on human diversity because human evolution is characterized by small groups of individuals leaving their parent population and establishing a new group in a new location. So human populations in different locations have come from many small founding populations throughout the world, throughout Europe, throughout Asia, etc. And most of these founding populations were established in the last 10,000 years or so. So as an example, Iceland was founded by a few thousand Vikings in the 9th and 10th century, and then were completely isolated for about 1,000 years until World War II. So in 1,000 years, they went from a few people to 270,000. If you look at the Icelandic population, there are alleles in that population that are common polymorphisms in Iceland, but are very uncommon in other populations. Now that's a reflection of that original founding Viking Viking population. And then because they are isolated, they took their own evolutionary trajectory. The people in in the rest of Europe took their own trajectory. So there are polymorphisms in Europe that are found there, but not in Iceland. The Icelandic population has also been subject to a variety of bottleneck effects. So founder effect made them different from the rest of Europe to begin with. And then there were 3 bottleneck effects. In the 15th century, the Black Death killed about 60% of the population. 1707, smallpox killed about 33% of the population. And then in 1783, a volcanic eruption killed about 25% of the population. So the founder effect plus all of these bottleneck effects makes them a very homogeneous group of people with very little variation. But they also have identifiable characteristics. And you have those same concepts that have characterized human populations throughout the world. So each region of the world has individuals with very specific recognizable characteristics, very specific alleles that are unique to those populations, and that gives them their own distinct phenotypes. It makes them identifiable based on their heritage. So people of Icelandic descent are going to have similar characteristics to the early Icelandic settlers. People from Africa are going to have more similar characteristics to other individuals in Africa, and so on. So our last Hardy-Weinberg assumption is that there's no differential reproduction. This means basically that no individual is surviving specifically because of the allele. In other words, there's no natural selection happening. So natural selection is where individuals survive and reproduce at a higher rate because of a particular trait. So we can call this differential reproduction. We can call this natural selection. Either one is fine. But whatever you call it, it changes allele frequencies. If one allele is more advantageous than the other, it's going to accumulate in the population while the other one decreases. So this graph here shows selection for allele A. Allele A is advantageous, so individuals with that allele are going to leave their offspring more frequently. So individuals who are homozygous for A are going to produce 6 offspring. So each homozygous individual leaves 6 homozygous individuals. Individuals who are heterozygous in this depiction are slightly less fit. They have slightly lower reproduction rates, and they're going to produce 4 offspring. Some of them are going to be homozygous, some of them are heterozygous. And then individuals who are homozygous recessive are the least fit. They have the least reproductive success because of their alleles. They're going to leave the least number of offspring. And so over here, if we start with an equal number of alleles in our population, but the blue alleles are more advantageous, it's going to slowly increase over time. Now, eventually, if this continues, the big A allele is going to be fixed in the population. So if that big A allele is going to become fixed in the population, how or why does genetic diversity even exist? Because that selection for one allele over the other should predict that that allele become fixed in the population and the less favorable alleles should be eliminated. There's an explanation for this, why there's genetic diversity at all then. So one explanation is that that favorable allele just hasn't become fixed yet. There hasn't been enough time for fixation to occur. There hasn't been enough generations. It could also be that genetic drift and migration are keeping that little a allele in the population. Individuals are coming in with the little a allele, keeping that frequency higher, or genetic drift has sort of selected for or increased that recessive a allele. Another explanation is that individuals are a product of their entire genetic makeup. So even if one allele is advantageous, there are other alleles working in that individual to affect their fitness. So their fitness is not solely dependent on that A allele. So there might be another allele that is even more advantageous. So it doesn't matter if that individual has the little A allele, because some other gene somewhere else is also affecting their reproductive rate. Maybe it's causing an increase. Next reason that there might be the next reason that genetic diversity might exist is that the environment is always changing. So one year a particular allele might be favorable, the next year it might not be so favorable, and so it decreases a little, and then in a later environmental change it becomes advantageous again. So the environment, how much selection pressure you're having on a particular allele can change over time. And then, of course, there might be other forms of selection at work. Again, we might be selecting for other alleles. There might be other Hardy-Weinberg assumptions that are operating on this population. For many traits, And in most populations, several of these explanations are going to explain why genetic diversity exists, why we still have a combination of alleles if one is more advantageous than the other.